Hi everyone. In this video, we're going to discuss chapter 1. So to start off with, you may be asking yourself, why do I need to study microbiology? So to start off with, when we think of the term microbiology, it can be broken down into 3 parts. Logi is the study of, bio means life, and micro means small organisms. And when we think about these small life forms, there's a big relationship between humans and these organisms. And for some of you, you may be going into the nursing program for physical therapy and maybe other biological or health medical fields. So it's very imperative to understand those relationships that humans have with microorganisms. Not all relationships with microorganisms are bad. We think about microbes in a very negative connotation. We think about germs and disease, but some can actually be beneficial and some are just harmless. So this is a very important understanding. We also have to understand infectious diseases. So the microorganisms that do cause disease and do cause problems. And the big question is, how do we prevent these infectious diseases from happening? And what are some possibilities and some mechanisms that we can use to treat? All right, so again, when we think about the term, what is microbiology? Microbiology is going to be the study of small life. And in fact, the best definition is understanding organisms that are too small to be seen with the naked eye. So because of this, a microscope has to be used. And so when we look at these pictures of microbes, we're going to see lots of different types. We're going to be discussing some prokaryotic organisms, such as bacteria and archaea. We're also going to be making mention of different fungal microbes, some protozoa, or what we call protists, which consists of algae and protozoa. And we'll also be discussing some worms as well, which are called helminths. So again, when we think about the term microbiology, it's the study of living organisms that are too small to be seen by the unaided eye. So a microscope has to be used. And again, when we think about the idea of microbes, we tend to think about the term germs. And there's a lot of misconceptions about that. Not all microorganisms are bad. In fact, 99% of them are harmless, and there's even some that can be beneficial. We're going to discuss microbiota, or sometimes formerly known as flora. And these are microorganisms that reside on your skin, in your mouth, in your GI tract, and they can even be beneficial. A lot of them cause competition, and so they will help to sort of inhibit bad organisms from growing. And so when we think about bacteria in particular, a lot of them are good or at least harmless, where they don't cause any problems. However, we do have some bacteria in particular that can be harmful. And if you hear the term pathogenic, I want you to think of patho as disease. Anytime you see the term path, it's going to be relating to disease. And genic, think genesis or beginning. These are going to be the organisms that can cause disease. And so you may hear the term pathogen. This is an agent that can cause disease, such as the coronavirus causing COVID-19. When we think of mono, which is infectious mononucleosis, that's due to the Epstein-Barr virus. Or Bordetella pertussis is going to be causing whooping cough or pertussis. So those agents are pathogenic. They can cause disease. But again, we're going to help to kind of slaughter some of those misconceptions about germs and start thinking about some benefits and not just the bad. And like I said before, we use the term flora. So flora is actually kind of an antiquated term. We don't use flora really anymore, and instead we use the term microbiota because it helps to actually classify lots of different organisms. Flora is really more classified towards plants, versus microbiota will actually help to make a more broad term for all different organisms. But when we talk about microbes that are present in or on the human body, we discuss them as normal microbiota. You may also hear the term resident microbiota. And again, that means that they are going to be presented in the body or they're going to be outside of the body. So when we think about normal microbiota, these can be all different types of organisms. On your skin at this moment in time, you have Staphylococcus epidermidis. You also may find some yeast cells. We may find even a couple protozoa as well that are associated with the body. So again, they can be on our skin, in our nasal passages, in our ears, in our mouth, in our GI tract. And so again, we're going to find them in multiple different areas. And these guys can be beneficial. They can cause benefits for us. So we would say that we have a mutualistic relationship. They can also just not cause us any harm, and we call this commensalism. Now, when we think about the benefits towards microbiota, they're actually our first line of defense against unwanted or pathogens because they're going to prevent growth of those pathogens. If you think about it, these microbiota are there to stay. They become a resident on your skin or in your GI tract. And they don't want any other competition coming in and taking their resource. So they're going to help to prevent growth of those unwanted guys, kind of create a check and balance for us. And again, when we think about their abundance, we're going to find about 2 million microbes per square inch on our skin. And in fact, when we start to discuss the actual kind of breakdown of how many cells we have, we're about 90% covered in microbes, and we're only about 10% human cells. So there's quite an abundance that we're going to find of microorganisms. In fecal matter, we find about 100 billion bacteria per gram. And if we think about the soil, there's about 10 million microbes per gram as well. In the air, you're going to find 50 to 100 microbes per cubic foot. And again, when we think about humans, with 100 trillion cells that are going to be on or in the body, only 10% are actually eukaryotic or your cells, what we consider to be animal cells. The rest is going to be microbial. So that could be bacteria, that can be a fungus, or again, that can be some sort of protozoa. All right, so when we think about how microbiota are going to develop, this starts right after birth. So normally when we think about someone being pregnant, there's these placental membranes that are sort of covering the newborn or the fetus at this point, and that is going to be a sterile environment. But the moment those placental membranes rupture and the baby is now going to be exiting the body, it's exposed to a world of microorganisms. As we said before, microbes are everywhere. They're going to be in the air, they're going to be on your skin. So from passing through that vaginal canal, from being kind of touched by different healthcare workers, such as nurses and doctors and parents, grandparents, and even from mom breastfeeding as well, there's now going to be this establishment of that microbiota. So within less than 12 hours, bacteria will actually colonize in the skin, on the skin, or in the body as well. So right off the bat, a baby entering the world is going to be exposed to microbes, and they're going to start to have that colonization take place. Now, there are some areas that should never have microbes and should be free or sterile in this circumstance here. There are many to discuss in your book. In fact, there's actually going to be a little chart on page 3 if you're looking through your manual. So just some areas to make mention of. If we think about the lower respiratory tract, such as what you would see in the bronchioles or alveoli, where there's true gas exchange, those should be some sterile areas. When we think about the upper reproductive systems of females, so again, thinking more along the lines of the fallopian tubes, the ovaries, those areas should be sterile. Especially upper kidneys as well, or upper urinary. So thinking about the ureters, the bladder, the cardiovascular system, so heart and blood vessels, the brain and spinal cord, the bone marrow, all of those areas should be sterile or free from microbes. And unfortunately, if you do find microbes in this area, it could potentially be an infection and it could cause issues later on. So we do have some areas that should not have microbes in the body, especially when we think about like blood or cerebral spinal fluid as well. Those areas should be free. But again, we're going to find microbes in specific areas on the body or in the body. All right, and like I said before, most organisms are really not a problem, but there are some that can be what are known as opportunistic pathogens. So they're normally not going to cause problems in a healthy individual, but given the opportunity, they may be pathogenic. And again, when we think about patho, we think about disease. So when given the opportunity, they may cause that disease or cause an infection. Usually this is going to be in someone that is immune compromised, such as what we see with Staphylococcus aureus and MRSA infections. Or it could be where you see microbes enter a part of the body where they shouldn't belong. So again, thinking about Staphylococcus aureus on the skin, again, is not a problem. If you have intact skin, it's not working its way inside the body. However, if you are working with a puncture wound, if you are working with a burn or some sort of unintact skin, and that Staphylococcus aureus leads its way inside the body, this can actually lead to a potential fatal infection. That's where we hear the term MRSA, methicillin-resistant Staphylococcus aureus. All right, so again, when we think of the term pathogen, gen is the agent and patho is disease. So this is an agent capable of causing disease. So when we think about a virus, for instance, the measles virus will cause measles. If we were to think about Trichomonas vaginalis, this can cause a STD. Or if we were to hear about like chlamydial infections, those have pathogens that can cause that infection and potentially cause disease. And when we think about the difference between a true pathogen and an opportunistic pathogen is normally not going to cause harm. And again, doesn't normally cause harm in a healthy host. However, if the person's immune compromised, or again, that microbe ends up somewhere where it really shouldn't, then it can potentially cause that infection and cause disease. But a true pathogen will always cause disease. If you're exposed to the influenza virus, you would develop influenza. So I hope that makes a little bit of difference and makes some sense. All right, so we have to worry about infectious diseases. And when we hear the term infectious disease, we're going to be thinking about diseases that can basically be contagious and be spread from host to host. So when a pathogen overcomes a host resistance, disease is going to happen. That pathogen has to work its way inside of the body, actually get through portals of entries, kind of actually work against our defenses, and then colonize and replicate, and then therefore cause this infection and then cause disease. So this may be a little different than other diseases that we see. In fact, some diseases are non-infectious. So you may have some diseases that are related to a pathogen. You may have some diseases that are non-infectious. So we will use the term communicable diseases for usually those contagious or transmittable diseases, and non-communicable for those that aren't. Now, one big issue in microbiology is we do have emerging infectious diseases coming up. So these are new diseases and new cases that are increasing in their incidence. So we're just seeing new emerging cases pop up. And some examples are those MRSA infections, again, those methicillin-resistant Staphylococcus aureus infections. We see issues with AIDS. This is due to the HIV virus, and AIDS is the syndrome based upon having HIV. And then E. coli, in particular, this strain, this O157H7 in particular, would be considered an infectious disease, an emerging infectious disease. Not all strains of E. coli are dangerous. In fact, actually, E. coli is part of our normal flora, but this particular strain can be actually fatal. All right, so thinking about that MRSA, again, this is methicillin-resistant Staphylococcus aureus. This is a bacterial infection, and it causes a wide variety of infections, but in particular, it causes a series of skin infections to take place. So just a little bit of history for it. In the 1950s, they started to see that penicillin resistance started to develop. What this means is that it's not the person being resistant against the antibiotic, it is the strain or the type of bacterium that is resistant against penicillin. Penicillin is a drug that would be given in combat to a bacterial infection, and unfortunately, this MRSA is going to be resistant. And then in the 1980s, we started to see this uprise of methicillin resistance. This is another antibiotic. And then finally in the 1990s, the MRSA resistance actually led to what is known as vancomycin being reported. And vancomycin is sort of the last resort drug given to be administered for a MRSA infection. It's a very difficult drug to take and has a lot of unwanted outcomes, but it's sort of the last chance for a really bad MRSA infection. All right, like I said before, when we think about E. coli, this is called Escherichia coli. Sometimes you also may hear it called Escherichia coli. And again, E. coli itself is actually part of our normal flora, but there's this particular strain that's a toxin-producing strain. It's the O157H7 in particular. And it was first seen in 1982, and it caused this huge issue worldwide with bloody diarrhea. And with this diarrhea, it led to issues with fluid disruption, causing problems with water balance, electrolyte balance, and again, it was potentially fatal for some. And then, like I said, we also have to worry about AIDS as well. AIDS is actually the syndrome. It's called acquired immunodeficiency syndrome, and it's caused by the HIV virus, the human immunodeficiency virus. The HIV virus attacks what are called CD4 helper T cells. We're going to discuss that in way more detail, so you don't have to worry about that right this moment. But it was first identified in 1981. And it originally started off as a worldwide epidemic, and it affected about 33 million people. And there's about 7,500 new infections every day. And now it's not considered an epidemic, but a pandemic, because it's not just in the United States, it's actually worldwide spread that we've noticed. So epidemic is not technically the correct term anymore. We would now call it or classify it as a pandemic. But it's a sexually transmitted disease affecting both males and females. Usually we'll see it with that direct contact with sexual use, but it can also be blood products and sharing of needles as well. But again, AIDS is the syndrome, the virus is HIV. And then we do have some re-emerging diseases. These are diseases that were kind of old diseases, but that are now starting to come back in highlights as well. A lot of problems with re-emerging diseases is due to the increased travel. So again, especially in the United States, we have kind of eradicated some of these diseases, and now they're coming back because now we can travel to areas that we couldn't before, and especially densely populated areas. There's also going to be a little bit more hesitant attitudes towards vaccination, or even like a lax attitude, and that can lead to some of these old diseases popping back up. And also, we do have an increased population of those that are immunocompromised. So their immune system doesn't properly operate, and therefore it makes them more susceptible to these diseases. So this would be, for instance, like whooping cough, which is going to be from Bordetella pertussis, and whooping cough is pertussis. The bacterial infection is called Bordetella pertussis, or the measles virus causing measles. We've now seen this kind of reemerging just recently, actually. All right, so again, getting into more detail about pertussis, again, the kind of known name is whooping cough. Bordetella pertussis is the bacterial or the bacteria that causes whooping cough or causes pertussis. And usually this is an infection of the lower respiratory system. And there is a vaccination before it. But before the DTaP vaccine was actually given, about 50% of individuals developed pertussis, and the vaccination really helped to decrease those numbers. But since the 1980s, we've seen lots of cases actually increasing significantly, and they'll infect adults and adolescents as well. And usually it's problems with not having an up-to-date booster. And again, unfortunately, even with those boosters, the immunity to the vaccine usually will decline after a few years, and there's really no protection after age 12 or by age 12. All right, so we're going to talk about some of the microbes in the environment here just for a moment. So these are what they kind of do and how they function in the environment. The first thing that we're going to make mention of is decomposition. So this would be like your bacteria and your fungi. They are going to be decomposers, and they're usually involved in sewage treatments, and they are going to help to break down lots of toxic waste, oil spills, other pollutants such as like even radiation as well. And so we call this biodegradation. They're going to help to be breaking down some of those components that normally, again, we would have it be toxic, but they actually can utilize and basically make them a little less dangerous, even like petroleum products as well. Now, with this being said, these organisms are also responsible for lots of food spoilage, actually billions of pounds of food spoilage per year. So this is what we think when we have soured milk or rotten vegetables, or even with canned food spoilage taking place. And so lots of pounds of foods every year are lost. So with this being said, again, that's lots of money that ends up being wasted. And we shouldn't throw just those rotten foods out in the garbage. In fact, we should actually try to make them into a compost and actually utilize them, because again, they can have that decomposition be very helpful. The decomposition processes help to actually break down some of those macromolecules and actually utilize them for plants. All right, so we also can think about element cycling, and this is very essential, basically for Earth itself, because they're involved in different types of cycles. We're going to discuss these cycles coming up in a couple chapters, but we're going to make mention of the carbon cycle, the nitrogen cycle, the sulfur cycle, and even the phosphorus cycle as well. We're also going to discuss photosynthesis in a little bit more detail of how some microorganisms utilize sunlight in order to actually make their food and utilize carbon. And we're going to be able to discuss this in a little bit more detail in a couple chapters coming through. All right, so this just kind of shows you some of those pictures again of those different element cyclings taking place. And like I said, we'll get into more detail about this later in the couple chapters. All right, we're also going to talk about food production. So we actually love food, and a lot of microbes are actually going to help to produce some of these foods. So some of our favorites are cheese, yogurt, bread, sauerkraut, vinegar, and even alcohol production is based upon utilizing microorganisms. In particular, beer. If you're a beer fan, you have to thank microbes for that fermentation process and helping to make some of these really yummy treats. All right, now we're going to get into a little history here just for a moment. And when we think about history, we're going to go back into time. In the 16 to 1800s, this was a great time of development for microbiology. And this is where kind of a lot of those influencers actually really led into the development and lots of debates. So prior to the 1670s timeframe, there wasn't a microscope and there really wasn't any knowledge about biology and microorganisms. So this led to lots of questions. And this led to some theories that eventually were disproved. So one of the terms that we're going to make mention of is spontaneous generation. And one of the questions that came up is spontaneous generation of microbial life possible? We also had to think about what causes disease. Prior knowledge was the idea that if a person was bad, if they had a bad character, or if they were poor, or if they didn't tithe enough to religious affiliations, they were considered to be a bad person, and then that disease would kind of be struck upon them. And then there was also this idea of, again, not really understanding how to prevent infections and diseases once you got it. So with this sort of change, and again, in this time frame, a lot of these questions became answered, and they've helped us to generate more ideas and more concepts behind the idea of what is disease, how do we prevent, and how do we treat. Okay, so starting off with the idea of spontaneous generation. Spontaneous generation is the idea that microorganisms can generate from non-living things. So if you had something dead, it could arise into something that was living. And this was a great debate for years. And the first person that really started challenging this theory was Francisco Redi in 1668. He was the first one that kind of said, oh, this doesn't really look right. And then him and someone named Louis Pasteur really went on to sort of truly debunk this idea of spontaneous generation. So that was kind of a major event that took place. We're going to talk more about Louis Pasteur coming up. But Francisco Redi was the first person that really challenged this idea of spontaneous generation, that, again, life forms could really uprise from dead or non-living material. Another big event that happened in microbiology was the creation or the invention of the microscope. And this was thanks to Anton van Leeuwenhoek in the 1600s. This was in 1673. Anton van Leeuwenhoek was actually a Dutch merchant, and he was the one that sort of understood or helped to create this knowledge of microorganisms. So he's a big person to thank for some of the advances that have happened in microbiology. Now, thinking about spontaneous generation again, just going back for a moment, there are some conflicting theories. The first thought was spontaneous generation, and this actually was proposed by Aristotle. And again, this is the idea that non-living organisms could arise living things. So again, you had this unseen life force that could cause life to arise. If you had a piece of meat arising from the dead were these maggots. If you had a dead person, arising from it could be bugs or something along those lines. And so it was the idea that non-living could arise living. And the conflicting theory to that was biogenesis. So again, I really like to break down terms to make a little bit more sense. If you see the term genesis, think of beginning, and then again, bio meaning life. And this is the idea that we now know is true. The spontaneous generation had then been debunked, but it took some time. Biogenesis is the idea that living organisms arise from pre-existing life, aka it has to be living to arise new life. And again, this is the theory that we now know to be true. But prior knowledge was the idea that spontaneous generation was the way that things worked. So this is where Francisco Redi comes in. He was the first guy that said, this doesn't make a whole lot of sense. So he decided to create his own little observation and experiment. And so in 1668, he filled 3 jars with decaying meat. He left one open, one with a mesh on it, and then he sealed one. And what he noticed was with the flask that was unsealed, you would start to see these maggots and flies. So again, that's kind of what had been known. And if you think back to the time frame, there wasn't proper storage of food and refrigeration. So this is probably what was seen quite a bit. And again, this is what kind of created that thought of spontaneous generation. But then what happened is when you see the sealed flask and the one that was covered with this gauze or this mesh, you also notice that there weren't any maggots on this meat. And again, that's what would have been expected because, again, it wouldn't matter that it was sealed, dead could arise living. Well, this is the first kind of thought of, okay, something doesn't make sense here. Why did one jar have a bunch of flies and the other 2 didn't? And as you can see, there's still flies on the top of the mesh, but they're not coming from the actual meat itself. And if you look at the flask that's sealed completely, there are not any flies, any maggots to be seen. Again, then we kind of have to go back and talk about Anton van Leeuwenhoek for a hot minute. So like I said, he was a Dutch merchant. He was from the Netherlands, and he was sort of known as the father of microbiology because he was the one that developed the first simple microbes. And what he did is he created his own little microscope. Now, if you look at this microscope, you can see on the left-hand side, the little picture of it, it was a very tiny microscope. There really wasn't a whole lot to it. He actually used a screw as the specimen holder. And although it's really kind of simple, it helped to see these first microbes. And this is how we really were able to discover the bacterial world, is thanks to Anton van Leeuwenhoek. So he examined water and he visualized all these tiny little animals. He would call them wee beasties or animalcules. And so that was kind of the first terms that were used. And what he was noticing were different fungi, algae, and what were called protozoa, these single-celled protozoa. And that's what you can see in the second image. So thanks to him and this sort of invention of the microscope, again, now we have very detailed microscopes. In lab, we will be using what is known as a light or compound microscope to see bacteria, but there are even more in-depth and way more expensive and complex microscopes that can even see details to electrons, these very, very tiny little components. And that's where you can see these really kind of beautiful, complex pictures of viruses and their spikes and just all this incredible detail. And it's thanks to this man here. All right, another person that we're going to make mention of is John Needham. So this is going to be in the 1700s. And again, he really was sort of thinking about this idea of microbial growth without sort of understanding what this kind of idea was. So what he did is he put boiled nutrient broth into these covered flasks. And so in the conditions, the nutrient broth was heated and then it was placed into a sealed flask. And the boiling was thought to kill some of this microbe, if not all life. And what he ended up noticing was there was still microbial growth. We're going to talk about detail about the different types of turbidity and kind of what was expected. But at this time, there wasn't any knowledge about heat-resistant microbes, aka microbes that could still withstand heat. So in his mind, he was thinking he would boil everything off. And again, from this picture, it shows that idea that microbes could still grow. So from this idea, it gave kind of a thought about and kind of criticizing this idea of spontaneous generation. And at this time, his thoughts were actually thinking that spontaneous generation was true. But A lot of critics really went against him because he didn't have a very clean environment when he did this. And it was suggested that microbes entered his flask in the air when he did transfer techniques. So, although he really thought and kind of was giving ideas to this idea of spontaneous generation, that was also disproved later on. And so the person that kind of really didn't like John Needham's sort of styles and techniques was this Lazzaro Spinazzelli. Like I said, he really criticized a lot of the techniques that were used. One thing is aseptic technique. If you expose your tubes to the air for long periods of time, microbes would be able to get in. And also, he didn't have a super clean environment when he did this. So he sort of repeated the work. And what he did is he boiled the nutrient solutions in flasks and then he immediately sealed them. And under these conditions, he actually noticed that there wasn't microbial growth. And in fact, it was clear. Versus the picture you see before, you can see that turbidity and you can see that microbial growth within the flask. For a Spinazzelli, you don't see that. It's clear. And so, looking at this idea, it really gave evidence against spontaneous generation and it supported the theory of biogenesis. Then finally, we get to Louis Pasteur. And Louis Pasteur is just an awesome guy in so many ways and shapes and forms. He has done so much for the microbiology world, the health field, and actually even for the food industry as well. If you've heard the term Pasteur, you may think of pasteurization, and this is the guy that helped with that. So in 1861, Louis Pasteur demonstrated that microbes were found in the air. And because they were found in the air, it debunked that spontaneous generation theory. So with Pasteur's experiments, what he did is he used a swan neck flask and he helped it to remain upright and there was no microbial growth that appeared. However, when the flask was tilted just a little bit, dust could actually get into the flask. And when the dust was able to get into the flask, it allowed for microbes to actually grow and become very cloudy in this area within just a few hours, if not a day. So when we start to see the pictures, this is kind of what happened. The Pasteur idea was that he was going to pour beef broth into this flask, and there were microbes that were present in the broth. Next, what he would do is he would heat the flask, and he would bend it into an S shape. And again, he boiled it for several minutes. So the idea would be that a lot of these organisms would actually be eradicated due to this boiling technique. And then the microorganisms didn't appear even after the cooled solution took place. And again, this bend in the neck really prevented any microbes from entering that flask. And there were no microorganisms even after long periods of time. So, depending on this situation, he really helped to debunk the idea of spontaneous generation because it showed that microbes were present in the air. All right, so with that being said, this leads us to the idea of the scientific method. So, debunking of the spontaneous generation and even the idea of the spontaneous generation debate really led to this idea of the scientific method. If you've taken other biology courses before, you have heard about the scientific method several times. So, what this is going to do is start with an observation. You have to see something, and then you're going to have a question behind it. So, you notice something about this physical world. You have a question about it, and then you're going to basically answer the question with a hypothesis. The hypothesis is your best guess, if you will, but it's supposed to be a logical explanation to answer that question. But we don't just leave it there and we don't take that hypothesis blindly. We have to actually test the hypothesis. And the hypothesis is tested through several experiments. The idea with experiments is to really either prove that your hypothesis is correct or disprove it, and then therefore come up with a new hypothesis. You don't throw out the question or the observation, but you try to answer it, and again, the best situation and the most logical explanation. So if you disprove the hypothesis or reject the hypothesis, you're going to have to go back and modify it. Again, then we would go back and test it through more experimentation. And if it becomes accepted, it can lead to what's known as a theory or a law. All right, so when we think about disease, we're going to talk more about Pasteur for a hot second. Louis Pasteur not only disproved spontaneous generation, he also developed the theory called the germ theory of disease. The germ theory of disease basically says microorganisms cause disease. If you were dealing with Bacillus anthracis, you would have anthrax. If you were dealing with infectious mononucleosis, it's due to the Epstein-Barr virus. Microbes cause the disease, aka the germ theory. And Louis Pasteur actually worked with Robert Koch, and Robert Koch helped to study the causative agents of disease. So what were those pathogens that were causing the disease? And he developed a very particular experimental step. It's called the Koch's postulates. And this is the way to directly link a microbe to that specific disease. That specific microbe causes that specific disease. And so Robert Koch actually demonstrated that anthrax was caused by Bacillus anthracis. So how Koch's postulates are really set up is in this sort of form and fashion. There's way more detail established, but this is sort of the nutshell approach. The first thing is that the microorganism must be found in abundance in all individuals with the disease, and it has to be absent and healthy. AKA, if someone has anthrax, you would find Bacillus anthracis in all of the people that are sick, and you would not find it in someone healthy. Next, the organism has to be isolated from the diseased person, and then it has to be grown in a pure culture in a lab setting. So if someone was to have Bacillus anthracis, the idea is that you could isolate that from the sick person and grow Bacillus anthracis in a pure culture in a lab. Then what would happen is once it's been grown in a lab, you're going to introduce that microorganism now to a healthy host. In this circumstance, a lot of times it was animals. So it was grown in a lab, isolated in a lab, and then it became inoculated into a healthy host. Now the healthy host is going to get sick with that disease. And then once that host gets sick with the disease, it has to be re-isolated, meaning they have to take a culture again, and they want to make sure that that host has that same microorganism. So again, it's going to be grown in a lab, and it's going to be grown in a pure culture. If you found that microorganism again, it really helped to seal the idea of the germ theory of disease. A specific germ, a specific agent caused that specific disease. Bacillus anthracis causes anthrax. Neisseria gonorrhea would cause gonorrhea. Mycobacterium tuberculosis would confirm tuberculosis. All right, so because of this work that Koch was able to do, a vaccine was actually developed. So it helped to the eradication of smallpox. It also helped to lots of declines for other microorganisms and diseases such as polio, the measles virus, and other childhood diseases. It also helped to create, again, this sort of establishment of the germ theory of disease. And these ideas were helped in the development of antibiotics as well. So again, a very big kind of hat tipping off to Robert Koch as well as to Louis Pasteur.